So we are ready to start the conference, the scientific part of it, and I welcome our first guest from foreign land, the land of promises, and from the Cornell University in New York, a very well-known and esteemed university in America academically. And you have been dealing with this disease for a long time. And now you will bring up two aspects about it, namely the role of physical exercise in this condition, and the other one is the role of the gut flora. Both two very hot areas in medicine, not only for ME, but for other diseases as well. So very much welcome, Professor Maureen Hanson.
Thank you. Is this volume okay? Okay.
Yes, here one.
Okay, so I was asked to talk about three different aspects of our work on ME/CFS, hence the divergence in the title. I did want to mention this report, if any of you are unfamiliar with it. This is really an excellent review of the literature. And one thing that's important for physicians who may not be familiar with the disease is that there is a clinician's guide. The clinician's guide and the entire report can be downloaded for no charge at this URL. You can just Google IOM and ME/CFS and you'll find that URL. I've had a number of patients tell me that it's helped to give this clinician's guide to their physician to learn more about the disease. Oops. So one thing I wanted to just start with was to emphasize the seriousness of the illness that I'm sure is known well to the many patients who must be in the audience. Many of you have probably done an SF36 questionnaire, which you answer questions that are then scored according to physical health and mental health and your social functioning. There was an important study done some years ago by Tony Komaroff, a leading ME/CFS physician in the US, and his colleagues that showed that individuals with ME/CFS this is very fast. Individuals with ME/CFS have worse condition than people with congestive heart failure or multiple sclerosis. I think that the higher the score you have, the healthier you are. So you see here 49, 62, and 70 for CFS versus heart failure versus MS. And likewise, the vitality of people with ME/CFS is much lower than people with congestive heart failure. This is jumping. That's just to emphasize the fact that this disease is really devastating, and we need to learn a lot more about it. There are a number now of documented biological abnormalities. I'm going to talk about three of these, which are the altered gut microbiome, the altered response to exercise, and the altered level of metabolites. But there are other ones that I'm not covering, such as the natural killer cell function, inflammatory proteins, changes in molecules in the brain. There's evidence for oxidative stress and hypometabolism and neuroinflammation. And also there's data on differences in gene expression in immune cells. So I'll start with the microbiome. There are as many microbial cells as human cells. Some of the earlier papers claimed there are 10 times as many microbial cells as human cells, but now it's generally recognized that that may be a slight exaggeration. The majority of these are in the large intestine, and these are important for enhanced nutrition and protection against pathogens. There are a lot of different factors that are correlated with gut microbiome composition. Your diet affects it, your health status, your genotype, and also your age. All of those have influences on your microbiota in your gut. The human microbiome is also, of course, associated with disease, and as more and more studies are done, more information is gained. So certainly obesity is researched a lot in the US, where we have a serious problem. Type 2 diabetes, Crohn's disease, ulcerative colitis, all those are diseases that are being studied with regard to the microbiome. Now, many ME/CFS patients complain of gastrointestinal symptoms, and one question is whether abnormal gastrointestinal function may be causing some of the inflammation that's seen in ME/CFS, and how does the gut microbiome differ from that of healthy controls? In a healthy gut, you have an effective barrier of your epithelial cells that keep out various pathogens and other molecules that would otherwise enter the bloodstream. But in an unhealthy gut, you can end up with some gaps between those cells that allow the unpleasant organisms and other molecules to get into your bloodstream. So this is thought to be one reason that you can have inflammation as a result of poor gut function. So the study that we performed was in collaboration with Dr. Susan Levine, a well-known ME/CFS physician in Manhattan. We had 30 female, 9 male controls, and 38 female, 11 male patients in the study. Their average age was about the same, but we had quite a range from 19 to 71. The body mass index was quite close, but we had quite a range from 16 to 47. Now, of these individuals who participated, we asked about various intestinal symptoms. We didn't specifically ask for irritable bowel syndrome, but we asked, if they had some unpleasant gut symptoms, and 8 of the 39 controls said they did, but of the patients, 32 of 47 said they had some gut discomfort of some sort. When we looked at the blood, we were able to see that there were higher levels of polysaccharides, which is a product that's made by certain types of gut bacteria. Not all gut bacteria make lipopolysaccharides. We could see that the average amount is higher in the ME/CFS than in the controls. And the protein, lipopolysaccharide binding protein that binds to LPS, it's a protein that the body has to detect the presence of LPS, is also higher in ME/CFS than in controls. This is probably the most striking increase there. And then also this protein, soluble CD14, detects the LPS-LBP complex. And it's another way that the bacteria detects that there's the presence of LPS in the blood. So this is consistent with the increase in inflammation that you might suspect as a result of the presence of LPS in the blood in a leaky gut. So there are other diseases in which elevated LPS has been reported. Fatty liver disease, HIV infection, Crohn's disease, and ulcerative colitis all have this finding of elevated LPS and LBP. So there's also a finding of lower diversity in the ME/CFS patient cohort. The way this is done is you obtain sequences of the gut microbiome. And the more sequences you obtain, you get more species. So you have to keep sequencing and sequencing, and then as you get additional sequences, you can detect some of the more rare species. So early on, if you only sequence, say, 5,000, get 5,000 sequences per sample, you won't really see much of a difference. But if you keep going till you get 30,000 sequences per sample, then you're able to see that there are actually more species present in the controls than in the patients. So there's a lower diversity of species in the ME/CFS patient cohort that we looked at. This is an indication of a loss of species richness in the ME/CFS microbiomes. Now, we also discovered that there were 24 genera of bacteria that were differentially abundant. These are all statistically significant differences. And we have some that are increased in the controls and some that are decreased in the controls relative to the ME/CFS. These two are decreased in the ME/CFS, and I'm going to mention a bit more about these groups in the circle there. These are known to be anti-inflammatory bacterial species. These are producing compounds that are considered to reduce inflammation. And the other aspect I would like to mention is that we could actually classify whether someone was ME/CFS or healthy at the 83% accuracy just by using the microbiome data and these blood assays in combination. So a combination of the LPS, LPB, and the soluble CD14, along with those species differences, we could identify 83% of the patients that way. So this work has been published in the journal Microbiome, and it's freely available if anyone wants to consult it. Since then, a group at Columbia has also done microbiome studies and has also found the same finding of differences in species between ME/CFS and controls. Now, there are some limitations of these microbiome analyses. We don't know whether a particular bacterial strain is differentially present in patients versus healthy individuals. So, for example, you've heard a lot about this dangerous E. coli strain that is present. While we, for example, would know that E. coli was present if there was a particularly bad strain of E. coli in the patients and the controls, the data that is obtained in this way would not tell us that. The other thing is that the data on bacterial DNA sequencing doesn't give you information about viruses. So I'll just briefly mention some early data we have on, that's not yet published, on the virome in ME/CFS. The viral component is largely bacteriophages and eukaryotic viruses. These viruses outnumber the microbial cells. There's about 10 to 1. And the majority are bacteriophages, which are viruses that attack bacteria. And these viruses, these bacteriophages, play a crucial role in modulating bacterial community structure and function. So that's a bacteriophage there attacking a bacterium. And they attack a bacterium and cause replication of themselves in the bacteria, and then that results in the bacteria being destroyed. They have an important role in maintaining inflammation in some inflammatory bowel diseases, ulcerative colitis and Crohn's disease. And in those diseases, there are more bacteriophages, and they result in a lower diversity of bacteria, as you might suspect, because of the bacteria attacking, the bacteriophage attacking the bacteria. So we undertook a preliminary study to examine the gut virome in the same samples that we analyzed before. This viral DNA is only 2 to 5% of the nucleic acids in there. So this, using standard methods for looking at DNA sequences, we examined the virome of our samples. Now, what we found was that there were some differences between the controls and the ME/CFS samples in these particular families of bacteriophage. Now, if we look more closely at these two down here, the cotovirales and the other category, we can see that, in fact, it's easier to see, looking close just at this group, this particular group, that we have more of this family and less of this family. And this increase here has also been reported in Crohn's disease and ulcerative colitis. Now, our sequencing wasn't deep enough to know whether or not we have eukaryotic viruses, human viruses in there that are differentially present. Our data currently is only sufficient to be examining the bacteriophage. So the summary of the gut viral microbiome is that we are seeing differences in ME/CFS versus controls. There are more viruses more types of bacteriophage in ME/CFS, and this is associated with this decrease in bacterial diversity. So it's possible that changes in this virome can be contributing to the intestinal inflammation and the bacterial dysbiosis, the lack of species richness. So one question is, what attempts, given that there are differences, what attempts are being made to alter the microbiomes in various diseases? And there have been manipulations of diet, of using prebiotics, which feed beneficial bacteria, probiotics, the bacteria themselves, antibiotics, and also fecal transplants. Something that I want to mention, because people will sometimes email me and say, should I take probiotics or prebiotics? We still don't really know the answer to that. So I just want to give an example of why you can't just unequivocally say, oh yes, I'll go out and take some probiotics. Here's a study in Crohn's disease, which is an intestinal inflammation disease, in which some of the treatments actually were harmful. So some of the probiotics actually made the patients worse instead of better. So it's not definite that probiotics are harmless and are fine to take, because they could actually make you worse. One reason that there need to be studies on probiotics and prebiotics in ME/CFS. Now, there is a study that is starting in Norway to look at fecal transplants to see whether they're effective in ME/CFS. We are actually collaborating with them. Peter Holger Janssen is leading the study, and he's planning to have 74 ME/CFS patients in the study and who will obtain fecal transplants, and then he'll be looking at the change in the microbiome from what they had earlier to after the transplant. And our role in this is going to be to find out if this affects this apparent leaky gut, whether we see reduced amounts of liposaccharide binding protein and soluble CD14 after the fecal transplants have taken hold, and it's been some months afterwards. But all sequencing of the gut microbial DNA and all of the characterization will be done by the groups in Norway. Okay, so now I'd like to turn to my next topic, which is post-exertional malaise. Now, post-exertional malaise is considered a hallmark symptom of the disease that is required, in fact, for the diagnosis according to the Institute of Medicine clinical diagnostic recommendations. So, in addition to the substantial reduction in activity and unrefreshing sleep, you're required to have post-exertional malaise to be diagnosed, and then either cognitive impairment or orthostatic intolerance. I was struck by Jose Montoya giving a talk recently that I saw on YouTube, in which he said that all of his thousands of patients fit this diagnostic criteria. So there was a very nice definition, I thought, of post-exertional malaise in this book, the IOM report, it's an exacerbation of a patient's symptoms following exertion that would have been easily tolerated prior to becoming ill. I think that's an important point. If you've always been unable to tolerate exercise, that's a different situation. So this exacerbation often occurs within 24 hours of the exertion, and patients often reach a steady state that they can tolerate. And for some people, that is being bedbound. They can't get out of bed. Other people can be up for a few hours, but not go out of their house, while others can work part-time. But very few people with the illness can work full-time. So one way to assess your aerobic capacity is to do a cardiopulmonary exercise test. And these tests have been going on in our collaborators' clinic for some time. And she uses, this is Betsy Keller at Ithaca College, and she uses a bicycle because it's too hard for the patients usually to use a treadmill. And too dangerous, actually, in case of falling over. This measures, during this test, you can measure the maximum heart rate, oxygen consumption, ventilatory threshold, maximum workload, and the respiratory exchange ratio. I'm going to explain those briefly. These CPET results, I'll refer to these tests as CPETs, are widely accepted as valid, and they've been standardized as to how you perform them by these various American societies. I imagine there's a comparable Swedish society that has standardized CPETs as well. In healthy individuals, the reproducibility, if you do a CPET one day and then you come back the next day and do it again, the reproducibility of the data is about 6 to 7%. So, for example, your heart rate would be about the same the next day, or your other figures. Now, what happens to ME/CFS patients was first observed by this group that we're also collaborating with, Mark Van Ness, Chris Snell, and Stacy Stevens, and they found that when patients performed a second CPET 24 hours later, after it was induced by an initial CPET, the post-exertional malaise was induced, their CPET values weren't the same, they were worse than 6 to 7% different. So this is in contrast to patients who've been tested who have heart failure, aortic aneurysm, renal disease, and lung disease. They can reproduce with all these very bad diseases, they can reproduce their CPET results. So whether there are other diseases that we don't know about where people don't reproduce their CPET results, we don't know yet because it usually hasn't been studied, but it would be very interesting to find out. So jumps forward here. So the initial report of this second-day CPET has been confirmed in some other studies, one of them that we did with Betsy Keller here, as well as these other two. So this is a reproducible result in ME/CFS. Now, how do you know that on the second day, it's just that the ME/CFS patients are being lazy and not working as hard as they should? That's an important question. The one thing that's always measured is the respiratory exchange ratio, and that's the amount of carbon dioxide exhaled over the amount of oxygen that's consumed. Now, if individuals are making their maximum effort possible, it's known that their respiratory exchange ratio is equal or greater than 1.1. So, as your effort increases, the muscles release more carbon dioxide and more oxygen is consumed, and that changes the ratio and increases it. So there's no way that any individual can deliberately alter the amount of carbon dioxide that they exhale. And so that's why this is considered an objective test. You can't manipulate the results by just not working as hard enough. So that's very critical for proving that there's something wrong when you have ME/CFS. This means the effort's comparable if your RER is greater than 1.1. Now, another concept that's important to know is the concept of VO2 max. So that's the volume of oxygen consumption per minute. And specific rates are needed to carry out specific activities. If you're jogging, you need to be using a higher oxygen consumption than if you were just sitting in a chair watching TV. So if your VO2 max is only the amount that is what's needed for sitting in a chair, then you obviously can't go out and jog. So that's why VO2 max is very important to find out. So this indicates the level of aerobic physical fitness and your functional capacity, your ability to do things like go out of the house or walk down the street. The ventilatory threshold is a way to measure when your anaerobic metabolism begins to be used. After a while, you have to switch to anaerobic metabolism, but the problem is it's less efficient than aerobic metabolism, and this also results in accumulation of lactic acid. And people can't sustain activities requiring this anaerobic metabolism. It's just impossible. You have a few minutes, but then you have to stop and recover. So the cardiopulmonary exercise testing was carried out by Betsy Keller's group at Ithaca College. The healthy subjects as controls are screened by questionnaires to identify people who don't exercise. And in the U.S., it's not hard to find people like that. And the patients are diagnosed by a physician who is experienced. And then the subjects did 2 maximal CPETs. So I want to give you an example of the data of a person who doesn't exercise in an active control. And this is the load, the workload on the bicycle. So it gradually increases from 0 up to 120. And the VO2 max then that you need goes up. So this person went from a VO2 max of just sitting on the bike of 3 up to 19. Their respiratory exchange ratio increased. See, it got up to 1.1, and by this time, the person couldn't go any further. Their heart rate went from 76 to 185. Blood pressure went up 110 to 160, and then in the second day, the VO2 max was very close to what it was in the first day. And if you look here, the heart rate, in fact, was only 1 beat off from maximum heart rate from the first to the second day. So the blood pressure also was about the same in the first and second day. So the VO2 max had a 1.8% decrease, and their anaerobic threshold was a 7% decrease. And that's within, as I said, the variability that you see when you test people who are healthy or have one of these other diseases. So here's an ME CFS patient. What happened to this person? Well, they worked as hard as they could. They got up to 1.1 on both days. But on the first day, they were at 25 for their VO2 max, but after post-exertional malaise, they decreased down to 19. So this person had a 25% decrease in their VO2 max, and their anaerobic threshold also decreased, a 27% decrease. So the heart rate was reproducible and blood pressure increased both days, but this person has a reduced VO2 max and anaerobic threshold. Now this individual would be considered, fairly fit with a rating of 25, but by the time they got to 19, they're actually falling into the range of being impaired. Here's an individual with an anaerobic anomaly. Again, their respiratory exchange ratio was fine. They got up above 1.1. And their VO2 max was about the same for both days. But they exhibited a decrease in their anaerobic threshold of 39%. So they started to use their anaerobic metabolism sooner on the second CPET than they did in the first, even though their heart rate and blood pressure was increased both days, and they had high RER, and reproduced their VO2 max. Now here's another interesting case. This person was able to exercised to 1.1 in both days, but for unknown reasons, their VO2 max actually decreased. But the reason that this decrease occurred is that for unknown reasons, their blood pressure wouldn't increase. So on the first day, their blood pressure went from 102 to 164, which is what you might expect for someone exercising. But on the second day, it just never increased. And as a result, they just couldn't pump enough blood into their body and had to give up at this point with much lower VO2 max, a 49% VO2 max. So why this is happening, we have no idea, but it's again a documentation of an abnormality. Now, at this point, Betsy Keller, shown here, has now tested 94 patients, and she's found that 34% of them have a physiological dysfunction. 39%, this is a VO2 max problem, 39% have anaerobic dysfunction, 43% have an autonomic dysfunction. Now, 29% of them did reproduce abnormally, so it's not an absolute diagnostic test for ME/CFS. Now, these people tend to be ones who have not been sick as long as others, and they also tend to be people who are very active and fairly athletic before they became ill. It's possible that these individuals will actually start developing these abnormalities as they can progress in the illness. So, how does metabolism differ in ME/CFS cases versus controls? We've published a study in which we looked at a very small group, a pilot group of 15 controls and 17 patients. And we were able to identify 30 metabolites that had differences at these statistically significant levels. And we found that the pathways that were affected were fat metabolism, energy and sugar metabolism, and amino acid and purine metabolism. These samples were extracted to examine polar metabolites using mass spectrometry, and 361 metabolites were identified. So 23 of these significant 33 metabolites significantly different were lower in the ME/CFS cases than in the controls. There were a few that were higher in the patients and the controls, but for the most part, the patients' amounts were lower than in the controls. And if we analyze what are the pathways that these metabolites correspond to, we find that, again, these are all the ones that were different. And it tends, again, fat metabolism, amino acid and purines, energy and sugar metabolism appear to be different. There's a major study that was done in Navio's group, and his group used a different platform to analyze the metabolites. He found he was able to examine 612 metabolites. We saw our platform only identified 361. But interestingly, this is just the data from females, because he did both females and males. We did only females. He had 37 metabolites decreased to our 33, and we had a number of common pathways that were decreased in both cohorts. These that he found that we didn't detect, and some that we found that he didn't detect, are partly due to the fact that we are using a different way of, we are able to identify different metabolites. So this disturbance in metabolites is not only these two studies, but there have been now a number of other studies that have demonstrated a difference in metabolites that you can find in ME/CFS. And this is a very active area of research that will be continued in the future by various groups to see if we can figure out what's happened to metabolism that could be reflected in this abnormality in the cardiopulmonary exercise tests and the post-exertional malaise. I'd like to acknowledge my collaborators. These individuals, with the exception of Betsy Keller and Ithaca College, are all at Cornell, although Jason now has taken a job at Duke. And since I seem to have a couple minutes left, I'm going to show you a couple more slides. Because I think you may be interested in our center. We have our Center for ME/CFS is called the Center for Enervating Neuroimmune Disease. And we like this name better than chronic fatigue syndrome because it really emphasizes some hallmark features of the disease, altered neurology, immunology, and the fact that people are enervated. They're feeling weak and lacking in energy. Now, as some of you may know, we were recently awarded one of the NIH ME/CFS Collaborative Research Center grants, which has 3 research projects, a clinical core and an integrative data analysis core. The first project is being led by Tacoma Shungu at Cornell Medical School, which is in New York City. Ithaca, New York, where I'm located, is 4 hours drive west of New York City. And he's going to be having people do MRIs and PET scans and having their blood drawn before and after exercise. So there'll be a PET scan before post-exertional malaise is induced, and then 24 hours later. The project that I'm leading is going to be looking at what's in blood, and we're going to be looking at metabolites. We're going to be looking at the release of extracellular vesicles. We'll have proteomics of proteins present in vesicles, and also the RNA that are present in vesicles released after exercise. Extracellular vesicle aspect of this project, we're going to be collaborating with Dr. Jose Alegra and Jesus Castro in Spain, although NIH doesn't allow us to send money from this particular grant to other places. We'll still find ways we can collaborate. Another project is being led by Andrew Grimson in my department at Cornell, and he will be sequencing, getting RNA data from single cells to see if we can find whether there's a particular type of immune cell that has abnormal levels of expression of particular genes. Because so far, people have been analyzing all of these together, and that will mask if there's a particular deranged type of cell in the bloodstream. We may be missing it by sequencing everything all together. So we're going to look to see if there's a particular kind of cell that has a problem. So the goals of the center research are to identify biomarkers, and there are 2 types of biomarkers that we really need. We want biomarkers for our diagnosis, and that this kind of biomarker requires specificity for ME/CFS. We need to know if it's for diagnosis that people with depression or some other disease don't have these markers. But we can also be interested in biomarkers for development of treatments, and these don't have to be specific for ME/CFS. So for example, there could be some difference between healthy patients and healthy people and ME/CFS patients that are also shared by, say, people with Parkinson's or other diseases. But if we know what those are, we can still see if they will improve, even if they're not specific to ME/CFS. If we can get those biomarkers altered back toward normal. The biomarkers are also important for new understanding of the pathophysiology of ME/CFS. And knowing more about the disease, we might be able to identify existing drugs that can improve patients' conditions. This will be the quickest way to make people better, is if we can find existing drugs that nobody has ever tried because we didn't know that that was important for ME/CFS. And of course, if we can find what processes are disrupted, then we'll know which processes need to have new drugs developed in order to improve the condition of people with ME/CFS. Thank you.
Thank you very much, Maureen, for this lecture, which contained a lot of information. I think we may have time for 1 or 2 questions in relation to the presentation. And if I may ask you first, you mentioned in the first part of your speech that the gut flora is extremely important here, and the bulk of that flora is found in the large bowel. How come that you can surgically remove the large bowel without affecting the general health in general of a human being? And the stool formation, yes, but the general health is unaffected by removal of the large bowel.
Yeah, that's an interesting question. It would be very interesting to study people who've had the large bowel be removed to see how that might have affected their health. I actually don't know how much data there is on what happens to individuals who've had, that, for example, what happens to their metabolites. Because some of the metabolites that we're going to be able to measure in the blood or metabolites are actually produced by gut bacteria, not just those that are produced by your own body. So that would be a very interesting experiment, actually, to look at people who've had the large bowel removed to see if their metabolites are significantly different. But that's not understood. No.
Okay. Any other short question? Yes, please.
Yes.
Yeah, I'm Lars Berman. I'm a retired bacteriologist.
I think your microphone doesn't work, does it? Now it will work.
I am Lars Berman.
No.
Can you hear me?
Well, I
I'm
can.
a retired bacteriologist. I was very interested as a bacteriologist in your the first speech. I just say good luck with the transplants, because that's the only way you can change the bowel flora. Secondly, I would like to mention that the ME is practically all bacteriology, because the mitochondria that carries on all the energy metabolism are actually bacteria that has invaded our cells during evolution. Secondly, all other metabolisms in the bacteria in our cells are actually copies of bacterial metabolism. So, for instance, you named purine metabolism that is carried on by the so-called one-col metabolism in human cells that handles methyl groups, and that is one of the problems of these patients. And this one-col metabolism is catalyzed by vitamin B2, 6, 9, and 12. And there are quite some data from a group in Göteborg in Sweden that you can that is a therapeutic way, because there is almost no other ways. But the pharmaceutical companies are of course not interested in vitamins, because there is no money. They are interested in these monoclonal antibodies because they are extremely expensive. So I just would like that you could find money to do studies on B vitamins, because it's cheap therapy and there are positive results from Sweden. Thank you.
Okay. I actually recall that there is someone doing a B12 study. I don't remember who it is, but there is someone doing a study of B12 in ME/CFS. I don't remember who it is, though. Do you remember? But maybe we'll be seeing that study soon. It looks like there's one other question over there.
Okay, one more question. Where is that? Okay.
I'm also interested in the pathology of the gut flora, and you told us that there was 2 bacteria that were anti-inflammatory, but which one were they?
It was Bifidobacterium, and I actually don't remember the other one. I'd have to look it up at my own slide. Whether the problem with a lot of the gut bacteria, and the reason you have to use the fecal transplant, is that we can't actually culture many of these bacteria. So, when you take a probiotic, let me see if I can get back there. When we take a probiotic, you're actually taking bacteria that can be cultured. So we're unable to actually replace most of the bacteria that are present. So, those are ones that you can't actually replace. So these are the genera that are decreased in the patients. The Ruminococcus, fecal bacterium, and Bifidobacterium.
I think there will be another way than fecal transplantation in the future. It is studied now in the south of Sweden to give kind of probiotics intra to colon through lavemang, and then you can put together the kind of mix that you like, that you prefer, that you think is good for changing the flora.
Right. As I mentioned, one of the problems with probiotics that you can give is the fact that a lot of these can't be cultured, that we have no way to grow them. So one thing I learned about at a meeting I attended on Saturday in Washington, D.C., is that there are actually now companies that are trying to do fecal transplant by pill, so that in the future you might actually be able to get a prescription for a pill that you can then take that would contain a lot of these bacteria. The source, of course, being donors, not cultures that are normally grown for probiotics or that are, for example, in yogurt.
Okay, thank you, everyone. We cut this discussion now and we go to the next speaker. And we'll have a general discussion, hopefully, at the end of the session here.